Good day, this is Professor Will, CBMD PhD. Today, Sunday, March 1st, 2020. It is 11:48 and 30 seconds p.m. Eastern Standard Time. And today I'm going to be doing a reading on scientific research. So let's get started here. How resident microbes restructure body chemistry. Date February 26, 2020. Source University of California, San Diego. Summary. A comparison of normal and germ-free mice revealed that as much as 70% of a mouse's gut chemistry is determined by its gut microbiome. Even in distant organs such as the uterus or the brain, approximately 20% of molecules were different in the mice with gut microbes. The makeup of our microbiomes, the unique communities of bacteria, viruses, and other microbes that live in and on us, have been linked with varying degrees of evidence to everything from inflammatory bowel disease to athletic performance. But exactly how could such tiny organisms have such immense influence on a person? University of California, San Diego researchers have created the first ever map of all the molecules in every organ of a mouse and the ways in which they are modified by microbes. In one surprising example, they discovered that microbes control the structure of bile acids in both mice and people. When you change the structure of molecules such as bile acids, you could change how cells talk to one another and which genes are turned on or off at a given time. And that might have huge consequences for body function and development of diseases. We hear a lot about how our own human genes influence our health and behaviors. So it may come as a shock to think that we could have molecules in the body that look and act the way they do. Not because of our genes, but because of another living organism. Mapping molecules and microbes in mice. The term compared germ-free sterile mice and mice with normal microbes. They use a laboratory technique called mass spectrometry to characterize the non-living microbes in every mouse organ. They identified as many molecules as possible by comparing them to reference structure in the GNPS database, a crowdsourced mass spectrometry repository developed by researchers and collaborators. They also determine which living microbes co-locate with these molecules by sequencing a specific genetic region that acts as a barcode for bacterial species. In total, they analyzed 768 samples from 96 sites at 29 different organs from 4 germ-free mice and 4 mice with normal microbes. The result was a map of all the molecules found throughout the body of a normal mouse with microbes, and a map of molecules throughout a mouse without microbes. A comparison of the maps revealed that as much as 70% of a mouse's gut's chemistry is determined by its gut microbiome. Even in distant organs such as the uterus or the brain, approximately 20% of molecules were different in the mice with gut microbes. Bacteria modify bile acids. After constructing these maps, the researchers homed in on one particular family of molecules that appeared to be significantly different when microbes were present, bile acids. Bile acids are primarily produced by the mouse or human liver, and they help digest fats and oils. They can also carry messages throughout the body. The team discovered bile acids with previously unknown structures in mice with normal microbiomes, but not in germ-free mice. It has long been known that host liver enzymes add amino acids to bile acids, specifically the amino acids glycine and taurine. But in mice with normal microbiomes, the researchers found that bacteria are tagging bile acids with other amino acids, phenylalanine, tyrosine, and leucine. More than 42,000 research papers have been published about bile acids over the course of 170 years, researchers say. And yet these modifications had been overlooked. Influence on human health. Curious if the same types of microbe modified bile acids are found in humans. The researchers used a tool they created, the Mass Spectrometry Research Tool, MASST, to search 1,004 public datasets of samples analyzed with mass spectrometry. They also analyzed by mass spectrometry approximately 3,000 fecal samples submitted to the American Gut Project, a large citizen science effort based at UC San Diego School of Medicine. Here's what they found. The unique microbial modified bile acids the researchers saw in mice were also present in up to 25.3% of all human samples in the dataset. These novel bile acids were more abundant in infants and patients with inflammatory bowel disease or cystic fibrosis. One way bile acids can deliver messages from the gut to other parts of the body is through specific gut receptors called farnesoid X receptors. Bile acids bind and activate the receptors, which then inhibit genes responsible for making more bile acids. The receptors also help regulate liver triglyceride levels and fluid regulation in the intestines, making them important in liver disease and possibly obesity. Several drugs are currently being developed to treat liver disease by activating farnesoid X receptors. Sure enough, in mice and human cells grown in the lab, researchers found that newly discovered microbe-modified bile acids strongly stimulate farnesoid X receptors, reducing expression of genes responsible for bile acid production in the liver. The study raises many questions about the role microbes might play in driving liver and other diseases, and in influencing activities of therapeutics such as drugs that target farnesoid X receptors. This study provides a clear example of how microbes can influence the expression of human genes, researchers say. What we still don't know is the downstream consequences this could have, or how we might be able to intervene to improve human health. What is a microbe? A microorganism or microbe is an organism which is microscopic, which means so small that people cannot see them with the naked eye. The study of microorganisms is called microbiology. Microorganisms include bacteria, fungi, archaea, protists, and viruses that are among the earliest known life forms. Which are the microbes? There are 5 main groups of microbes populating our Earth. These include bacteria, viruses, fungi, algae, and protozoa. You are most likely familiar with some of these organisms. What are the 4 types of microbes? Types of microorganisms: the major group of microorganisms, namely bacteria, archaea, fungi, yeast, and molds, algae, protozoa, and viruses. What is the difference between microorganism and microbes? They are same. Microorganism is the word used in plural, and microbe as singular. Plural dictionary differentiation. Microbes are microorganisms. Microbes include bacteria, viruses, fungi, and parasites. What are useful microbes? Useful microbes in human welfare. Microorganisms include bacteria, fungi, protozoa, some algae, viruses, viroids, and also prions. Some other microorganisms are beneficial and contribute to human welfare. Are microbes living? A microbe is any living organism that spends its life at a size too tiny to be seen with the naked eye. Microbes include bacteria and archaeobacteria, protists, some fungi, and even some very tiny animals that are too small to be seen without the aid of a microscope. How are microbes harmful? A few harmful microbes, for example, less than 1% of bacteria, can invade our body, the host, and make us ill. Microbes cause infectious disease such as flu and measles. Different diseases are caused by different types of microorganisms. Microbes that cause disease are called pathogens. Good day, this is Professor Will, CBMD, PhD. Well, this sums up my preface on today's research. Hope you enjoyed listening. Have a great day.